Hello learners, today we are going to learn about cell and blood. Well, a cell is the smallest self-functioning unit found in all living organisms. Cells may exist as independent unit in all organisms, may in the form of colonies or tissues as in the higher plants and the animals. Discovery of cells. In 1665, Robert Hooke discovered the cell. He observed a honeycomb-like structure in a cork. Cell theory. First point, all organisms are composed of cells and their products. The bodies of animals and plants are composed of the cells and the products of cells. All plant tissues are made up of cells. These cells are having thin outer layer which we now know as plasma membrane. The second point of cell theory is all cells arises from the pre-existing cells. Cells divides and new cells are formed from pre-existing cells. Louis Pasteur in 1862, based on his experiment, successfully established the life originated from the pre-existing life. Unicellular organisms and multicellular organisms. The single-celled organisms are called unicellular organisms, like bacteria, amoeba. These organisms are capable to breathe, divided, and nourish, while the organisms with more than one cell is capable to breathe, that is termed multicellular organisms. For examples, humans, birds, reptiles, and fishes. There are three major categories on the basis of levels of differentiation in the cells. One is undifferentiative cells, differentiative cells, and dedifferentiative cells. The next topic is structure of cell. We all know there are two types of cells. One, eukaryotic cells and two, prokaryotic cells. Eukaryotic cells, these are large multicellular cells, have a nucleus boundary by a double layered membrane. For example, all plants, all animal cells. They show a higher degree of differentiation. And the other side, prokaryotic cells are very small cells and multiply very rapidly. For example, bacteria cells and blue-green algae. Now we must have a basic idea that what is eukaryotic cells. These cells have highly complex organizations, consist a cytoplasmic matrix and other organelles such as mitochondria, endoplasmic reticulum, Golgi Apparatus, ribosomes, nucleus, etc. Plant eukaryotic cells have cell wall, but animal do not have the cell wall portion. Firstly, plasma membrane. Cells are guarded from outside by a membrane that is called plasma membrane. This membrane controls the flow of molecules to or from to the cells. The membrane is responsible for the communication a cell with the outsider world. This cell membrane is a dramatic structure undergoing various type of changes that's called fluid mosaic model. It is a two-dimensional structure. The membrane performs an incredible variety of roles in human body. The protein found freely on the face acts as transporter and serves to carry molecules across the cell membrane. The cell maintenance the cell environment. It also has some such important enzymes which are essential for the critical metabolic processes like respiration, photosynthesis, and lipid fat breakdown, etc. These membranes are selectively permeable, so they help in the transport of certain molecules. Next, cytoskeleton. The ability of cells to change its shape or carry well differentiated movements depends on these skeleton systems. The cytoskeleton is a network of fibers running throughout the matrix of the living cells and provides a framework structure. Cytoskeleton has three parts: microfilaments, microtubules, and intermediate filaments. Next, endoplasmic reticulum. The endoplasmic reticulum is a fine reticulum network on the plasma of the cell. The endoplasmic surface is studied on its outer surface, and ribosomes are attached on it. Those are called rough endoplasmic reticulum, and where the ribosomes are absent, that's called smooth endoplasmic reticulum. So there is a two type of endoplasmic reticulum. One is RER, rough endoplasmic reticulum, and other is SER, smooth endoplasmic reticulum. RER synthesis proteins and SER synthesis lipids. Next, Golgi apparatus. It is consists of stuck of flat membranous sacs. Next, lysosome. It acts as a scavengers of the cells and involved in the intracellular digestion. Those are releases enzymes and digested all the cell particles. If they are released in tissues, they may kill other cells and the cell in which they were stored. Thus, the lysosomes are called suicidal bag of human body. Cells are takes in material within that is called endocytosis, and when the cells give out the materials or excrete the materials, that is called exocytosis. Endocytosis is divided into two types. One is pinocytosis and other is phagocytosis. Next, ribosomes. Those are small but complex in structures, roughly 20 to 30 nm in diameter. There are two unequal subunits, one is large and other is small. They are in diameter. There are two unequal subunits, one is large and other is small. They are responsible for the synthesis of proteins. Next, mitochondria. It is called powerhouse of cells. They are associated in the generation of ATP, the energy currency of the body. Mitochondria has two membranes, one is outer membranes, other is inner membranes. The inner membranes has many infoldings, that is called cristae. Next is centrosomes, which is consist of centrioles and centrosphere. Centrioles are composed of some microtubules in the arrangement of cylindrical structure. Cilia and flagella. Those are the projections from the cell, and they are made up of the microtubules as centrioles. The last structure is nucleus. Nucleus is the core of the cell. It is the part of cell that contacts with DNA and RNA, and it is responsible for growth and reproduction. It is an organelle which controls all the activities of the cells. The nucleus is a storehouse of hereditary information. Two membranes, one nuclear envelope which is encloses. Next topic is tissues. There are four types of tissues: epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Epithelial tissues are generally closed, packed in cells. We can observe after taking scraping from the mucosa, we can observe the epithelial tissues. Epithelial tissues line external surfaces like skin, cavities like stomach, tubules like urethra, and produces secretions and proliferate. Epithelial tissues in gland exude secretions. For example, the earthworm may secrete a cuticle and grab may secrete a cell. Next is connective tissue. Areolar tissue, adipose tissue, white fibrous tissue, tendon, ligament, cartilage, and bone. Next is muscle tissue. Cardiac muscle, skeletal muscle, and smooth muscle. Nervous tissue. There are two types of nervous tissue cells. One is neurons and other is glial cells. Neurons have numerous long processes which is transmit the nerve impulse, and the glial cells have short processes that protects neurons. Next topic is blood and its composition. Firstly, the plasma. Plasma, as you already know, it is a fluid which is the cellular elements of the bloods. Plasma is a faint yellow colored and slightly alkaline in pH. Plasma is watery, 90% watery. There are three types of plasma protein. One is albumin, two is globulin, and three is fibrinogen. Those are maintaining the osmotic pressure of the blood and viscosity of the bloods and helps to exchange the fluid from the blood and capillaries. Other functions are protein reserve, antibodies creation, clottings, and transported. Blood cells: RBC or red blood cells, white BC or white blood cells, and the other is platelets or thrombocytes. RBCs, those are called erythrocytes. RBCs can swell quite bit before blasting the hypotonic solution. There is a bioconcave shape. RBCs occupy about 45% of blood volume. This figure is referred to as packed cell volume. Next is white BCs. Those are called leukocytes. They are colorless. There is many types of leukocytes or white BC like neutrophil, eosinophil, basophil, lymphocyte, and monocyte. Neutrophil is 50 to 70% in blood. Eosinophil is present 2 to 4%. Basophil is present 0.5 to 1%, and lymphocyte present 20 to 40%. Monocyte 3 to 8%. Lymphocytes have extraordinary divisions by their functions. One is B lymphocytes and two is T lymphocytes. Last is platelets. Main function of platelets is release the chemical substances called serotonin and aggregate the plug of the vascular plug, site of the injury, have like skeletal muscles acting and myosin proteins which contract to the bring clot formation. Next topic is erythropoiesis. It is derived from the erythros meaning red and the poiesis meaning making. It is the part of the broader process like hemopoiesis, that is formation of the blood cells in general. Regulation of erythropoiesis. There are some factors like erythropoietin, androgens, estrogens, thyroxines, neural controls, product of hemolysis, and dietary factors. Next is blood groups. According to the ABO blood group system, there are 4 differentiate blood groups like A, B, AB, and O. Based on the concept, O group individuals considered as universal donor and AB group individuals called as universal receptors. RH blood group system. Some individuals also have RH factor systems, which is positive, called RH positive, and who is not positive, it is called RH negative. This is the on basis of RH process. About 85% of humans are RH positive, but among the African, almost everyone's are RH negative. Sometimes RH complications leads to the generally not seen with the type of mismatched transfusion. If a RH negative person is given to the RH positive person blood, there is no immediate adverse reaction because RH negative individuals do not normally have anti-RH antibodies, which may damage the donor blood cells. By this time, most of the donor cells die a natural death. The anti-RH antibodies can do no harm, hence first phase of transfusion passes silently. If the second dose is given, the memory cells ensure anti-RH antibodies are synthesized in proper amounts. The secondary transfusion acts as a booster dose. High dose of anti-RH antibodies can be achieved donor cells as thus damage in that time. There are a critical manifestation called erythroblastosis fetalis. Erythroblastosis fetalis is besides the risk of the mismatch reaction during second or during the subsequent blood transfusion, giving RH positive blood involves in the risk in cases. It is a complication during pregnancy period. This is due to following reasons. Red blood cells of fetus are unable to cross normal placenta, thus has to be some abnormality in the placenta. Fetal red cells may be destroyed. Next topic is anemia. Anemia, as we already know, it means deficiency of RBCs due to some rapid loss or slow production of the RBCs. Some types of anemia and their physiological causes: hemorrhagic or blood loss anemia, aplastic anemia, iron deficiency anemia, megaloblastic anemia, hemolytic anemia. The next is morphological classification of anemia: hypochronic, hyperchronic, macrocytic, microcytic, normochromic. Macrocytic normochromic anemia is called pernicious anemia, folic acid deficiency, etc. Normochromic, normocytic anemia, acute blood loss, and hemolytic anemias. Microcytic normochromic, neoplastic anemia. Microcytic hypochromic is iron deficiency anemia. Hemostasis. It ensures blood to coagulate into a solid gel during injury and also ensures free flow of blood in the vessels under normal conditions. Hemostasis is the balance of physiological process which on one hand prevent excessive bleeding and other hand maintain a circulation by keeping the blood in an uncoagulated state. Hemophilia. Hemophilia is a disease characterized by excessive bleeding due to deficiency of clotting factor 8. The patient needs repeated blood transfusion or may need a factor 8 concentration. Blood transfusion. Therefore, it is refers to the effusion of blood to blood components in an individual's for the medical treatment like anemia, loss of blood due to injury, etc. Transfused blood may be homologous from a donor or autologous previous stored blood.